Namaste students, I'm Dr. Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veteran Science, Jayoti Vidyapeeth, Women's University, Jaipur. Today I'm going to discuss about the topic microbial growth. So this topic belongs to the subject fundamentals of microbiology, and the subject is related to microbial nutrition. So first we need to discuss about the microbial growth and what is microbial growth. And microbial growth involves an increase in the number of cells because there are some microbial cells, so they increase in number, and their increase in number is known as the microbial growth. Similarly, they also increase in their size also. So, but it cannot be said as the increase in the size of the microbial growth, but increase in number of the microorganisms is known as the microbial growth. So there are two levels of microbial growth. First, cells synthesize new components and increase in its size. Okay, first it will create it will synthesize its own DNA, RNA, proteins, and then then the cells will increase in their uh, size. Okay. The second thing is that after increase in the size, the number of cells will increase. Okay, so they will divide and they will increase in their number. Increase in cellular constituent that may result in the increase in cell number. Example, when microorganism reproduced by budding or binary fission is the example of the microbial growth. And similarly, increase in the cell size is also a part of the microbial growth. It is not can be so said that the microbial growth, but it is part of the microbial growth. Example, some microorganisms have nuclear division that are not accompanied by cell divisions. Some microbiologists usually study population growth rather than growth of individual cells. The bacterial division and reproduction is a mode for the bacterial microorganism growth, and bacteria normally reproduce by a method which is known as the binary fission. What is binary fission? Binary fission is the process of bacteria reproduction where one cell becomes two. One cell becomes two. So it is known as the binary fission. So will uh, one cell will divide from middle and it will create two equal cells. So it is known as the binary fission. Similarly, the generation time is also very important. What is generation time? uh, the time which is required for a cell to divide its population is called the generation time. The time required for a cell to divide its population is known as the generation time. Okay, so they will divide, and after division they will multiply, and this will be the time which is taken by these microorganisms is known as the generation time. For example, the E. coli takes 20 minutes for the and Mycobacterium tuberculosis takes 18 hours, and Mycobacterium leprae takes 14 days for the generation time, as their generation time. Okay. Now we talk about stages in the microbial growth curve. When a few bacteria have inoculated into a liquid growth medium, it is possible to plot a bacterial growth curve. Okay, so when they will multiply them, we can plot a bacterial growth curve. Okay, so there are four basic growth curve: the lag phase, log phase, stationary phase, and death phase. The bacterial population follows a sequential series of growth phase, the lag, log, stationary, and death phase, and then knowledge of bacterial growth curve is critical to understand the population dynamics and population control in course of the infection disease. So the graph is like this. So what? So it is sequential. In the lag phase, there is no increase in the growth. Exponential phase have the maximum rate of division and population growth. Stationary phase and that the population growth ceases, and death phase it is a decline phase. So we will discuss one by one these four phases. First lag phase, bacteria first introduced into an environment or culture media. So when we insert the bacterial culture for the first time, it will enter into the lag phase because everything is new here. So it will prepare itself. So, so everything is new in the lag phase because it is introduced for the first time. So it will prepare itself. It will prepare its own DNA, its own protein. So nothing is going to increase. So the cell will start to increase in their size. No, there is not any kind of microbial uh, microbial division. Okay. So bacteria are first introduced into an environment of media culture, and bacteria try to adapt to the nutrients. And lag phase, which appear to grow in size and synthesize enzymes etc. And it varies in length. Some cases it is short, and some cases it may be absent. In some cases it will be longer. Next is the low phase. When bacteria have entered into the environment, it enters to the lag phase. After that lag phase, a new phase, that is log phase, arrives because bacteria is now adapted to the environment. Okay, and it has increased in their size. So now they will increase in their number. They will divide continuously because the media is new here. Media is stress here, so they will undoubtedly they will utilize the media continuously. The stress media, so they will utilize it. So log phase is also known as the uh, exponential phase and log phase, and the the rate of growth is constant, and population number of cells undergoing binary fission, it doubles the constant interval, which is called the generation term, and it is the best phase for the microorganism to grow because the bacterial population increases exponentially in this phase. Okay? So everything is adapted to the microorganism. So they will continuously divide, and they will continuously convert themselves. The next phase is stationary phase. If exponential growth continue, okay, when we know that media was new for the exponential phase, but now the media is not new because all cells have been divided. No new cells are divided. So when the media has been utilized, and a lot and some of the cells which have been grown, which have been divided, will start dying. Okay, and some of the cells will continue to divide. So there will be uh, ratio, equal ratio of the cells which are dying and the cells which are growing. Okay, so the number of cells which are growing, which are dividing, and the number of cells which are uh, dying will be equal. So we will see a simple plot, constant plot. Okay, or in this stage microbial rate is equal to the microbial growth. So Death is equal to the growth. So we will observe a plain graph. Okay, straight. Okay, horizontal uh, line in the graph. The possible reason for entry into the extinction phase is due to the nutrient limitation, limited oxygen availability, and toxic waste accumulation, uh, critical population density risk. So these are the reasons. Again, a nutrient limitation, limited oxygen availability, toxic waste accumulation, critical population density. Okay. So these are the reasons by which the cell goes to the station phase. Next phase is the death phase, and this is also known as the decline phase, in which the there is a decline in the number of viable cells, and the cells start dying, and usually at exponential rate. So they will start dying at exponential rate. So the number of cells which are dying is very high. Okay, and death is the loss of the ability to produce some microorganism. So death is the loss of ability to produce, and in some cases, death rate slows due to the accumulation of the resistant cells, and it is slower than the log phase. Okay. Because some cells will still start to divide. So this is the death phase, and this is the final phase. So in this video, I discuss about the microbial growth and some microbial growth curve. So in the next video, I will discuss some measurement of microbial growth. So my lecture is over here. If you have any query, you can ask me. Thank you very much.